So the typical problem setting in which you are when you want to use quality by design is that actually you want to optimize synthesis reaction or any chemical or pharmaceutical process or product. And so you have actually critical quality attributes that characterize how well the process works or how well the product fulfills the requirement. And next to these quality attributes, which might be tablet hardness, the impurities in your product, for instance, or on an economical scale, which is not really quality related, to maximize the reaction yield. Actually, all these can be influenced by using appropriate values of process parameters, choosing the right materials, and also using the materials in the right quantities. This is actually what is called, in the context of quality by design, critical process parameters and material attributes, so CPPs and CMA. And this doesn't come from our company. This comes actually from regulations, and the classical regulations in this area for pharmaceutical development are ICH 28 from 2009 and the Process Analytical Technology Regulation from the FDA from 2004. Then, in this context, what these regulations tell you to do is actually to design efficient processes, which everybody would like to do, of course, and this to guarantee product quality. And mainly what it tells is that you have to put the specification, shall it be of the parameter or of the product attributes, based on a scientific understanding of the influence of the parameters, material attributes on the product quality, so it be a formulated product or just a chemical product. And besides this, these regulations, they say also you should supervise the quality in real time as the product goes into production. So this approach, actually the regulations, they precise that the process knowledge which is actually multidimensional, because you have several parameters that can together have an impact on the quality attributes. This, you can express that in form of a design space, and in order to get this design space, a recommended tool is already in the regulation statistical design of experiments. And it gets more and more that the authorities or the auditors ask whether you used it or not, how you got the design space, and so on. So far for the introduction and for explaining or refreshing everybody why we speak about design of experiments today in your context. So assuming that this is clear now, what I would like to do in the second part is to demystify design of experiments. You may be afraid of the word statistical because you don't like statistics at all, or you never liked it. And what I like to show here is what design of experiments is all about. And I'd like to do that as easily as possible so that it becomes clear to everybody. So mainly, you want to do experiments in order to optimize something, let's say optimize the yield. Then you have many possibilities to vary parameters if there are several factors or several parameters that play a role. What we have here is actually we have two parameters, the temperature and the concentration, and what we want to optimize is actually the yield. So since I cannot make any experiment today in this webinar, I have taken a contour plot of the yield, which is this picture here, that tells you that the best yield actually here, of course, when you do experiments, you don't know this in advance until you have done the experiments. So here, since we cannot measure the yield, actually we know that the best point is here. Then these lines, they show that, for instance, here on this line 85, the yield is constantly 85 and so on. So it's a kind of hill that you see from the top. And actually, as you walk in this direction, actually the yield goes down as you walk down the hill. So in this factor, in this one factor at a time method, which is widely used by the majority of the scientists, actually, also in the pharmaceutical industry, the idea is to optimize the yield by varying some factor first. So here we choose the concentration. And as you see with these blue points here, we make 6 experiments for different concentrations, keeping the temperature constant at 70 degrees. And then we get 6 measurements, of course, in the lab. And what one would see is that the second point here is closest, is the one with the best result. It's about 62%. You can see following the control lines. And so this one is the best result. So then one decides to keep that and to play with the temperature then by making 3 additional experiments. So in total we've done 9 experiments, 6 horizontally and 3 additional ones vertically. And we look out of these 9 results, what is the best result? And the best results are actually marked by these green points you see here. And if you follow the line, they are on the line 75. So the conclusion after these 9 experiments is that the best yield is 75. Obviously, you can see in this picture that the best region close to 90 or 85 has never been looked at. And this is due intrinsically to the method chosen, because you do one factor at a time, and this doesn't enable to look everywhere at the same time. So what would design of experiments do on the contrary to this? I will take exactly the same picture without the points, and we'll start again with a design of experiments method. So if you use design of experiments, the purpose is still to optimize the yield. The name of the design is not so relevant for the moment. And the main idea is that instead of varying one factor at a time, you vary everything simultaneously. So you vary both simultaneously, and then you get 9 points here as well. And these points, as you can see, they are not on 2 axes only, they are spread over the whole zone. Where these points are depends on the design, and this is calculated on the basis of statistical concepts. So if you look at this, what you see is that this point has a yield of 80, let's say 85 or 86. This one is also 80 something, and this one here has 75. So we already have 3 points that are better than what we believed to be the optimum according to the other method. So finally, we find the best combination, and the yield is above 85. The number of experiments performed in both cases is the same, it's 9. So as a summary, we could say experimental design, DoE, is more than experimentation. Actually, you do all the experiments needed, but no superfluous ones, especially if I go back to the previous picture, this one, you could consider that the 6 experiments made here horizontally, these are too many, and maybe 4 would have been sufficient. Then what we can see as well is that it's reliable because you look everywhere, so all the possibilities are optimally covered. And one essential point for getting scientific knowledge about a process or product is that the detection of interactions become possible. What interactions mean is that the effect of one parameter depends on the value of another. I'd like to show you that on the previous slide again. And if you do it like this, and if you take a temperature of 70 degrees and you increase the concentration, then you can see that the yield will increase up to here about, and then it decreases again. But if you do this for a temperature of 100 or 105, the yield will increase as you increase the concentration up to about here, and only then it will decrease. So it shows that the impact of the concentration on the yield is temperature dependent. It's not always the same for all temperatures. So if we go back to the summary now, interactions can be found, which is very valuable to understand what goes on. And finally, it's goal-oriented, systematic, it's not trial and error, and I don't encourage anybody to tell an auditor that you chose these factor settings because it worked and it was good enough. No auditor would be happy with that in the pharmaceutical industry. So to summarize, DoE is getting maximum information with minimum effort. Of course, you may say, yeah, this is good and nice to know, but my projects are very different, and in each company the projects are also very different. And this might be a challenge for the method that doesn't know what the practical case study is about. So let's discuss that now. The diversity of practical settings, can it be a problem or not? In fact, you have several situations that can occur. The number of factors actually can be large. If it's a new process, a new technology, you have no idea, and you have maybe 10 or 12 parameters that you consider that could play a role, but you don't know which ones are important. Or it's a well-known process that you want to optimize, so you know which parameters are important, but you want to do some fine-tuning of their setting. So this is the first area of diversity of the case study. Besides this, the types of factors can be very different. They can be continuous, like the reaction time, the temperature, steering speed, and so on. So this you may set to any value in a reasonable interval, of course. So all those are in general independently variable. Besides this, you may have discrete factors like the stirrer type, for instance. If you have like two stirrers and you have to choose one stirrer out of the two, of course, the variable is restricted because you cannot choose stirrer 1.5. You have to choose stirrer 1 or stirrer 2. And it's the same for selecting materials or selecting a solvent. Furthermore, the materials also play a role. So there you can choose another supplier. So it looks like the stirrer case. Or you can change the material quantities. So overall, it's similar to process factors. And if some of you work in the formulation area in the pharmaceutical industry or in any other industry, you may have to do with formulation of recipes, like finding the good proportion of each component in a formulation. And for this, you need a special type of parameters because the sum of the components of the formulation must sum up to 100% in general. So we have a very large diversity of settings, and in order for DoE, which is a pure statistical method, to work for that, you need a sound strategy. And this is the clue in applying DoE efficiently. We consider, as actually most people do, that a three-phase strategy is ideal. Imagine that you have a large problem with, let's say, 15 parameters, and of course everybody has proposed in your group that some parameter would be important or worthwhile considering. So if you take actually one parameter, out of them, you don't know, you have actually 15. So which ones are important? In order to sort that out, you need a screening phase which you can use, for which you can use DoE already, and it will help you to find out of the 15, maybe 6 or 7 or 8 that are more important than the others. The others were just good ideas, but don't turn out to be very important at all. Or maybe are not important at all or do not play a role. Assuming that out of the 15 parameters you have found 7 important ones, of course you want to look at these 7 in more detail. And this will be the next phase, which we call modeling, in which we analyze, for instance, parameter interactions, and this enables us, among the 7 parameters, also to find maybe 2 or 3 or 4 essential ones. So, in other words, to perform a further factor reduction. And finally, knowing that 3 parameters are essential, then you will focus on these ones to do fine-tuning, and this is what we call the third phase, optimization, where you'll find an optimal combination of the values of these factors. But not only the essential factors will be optimized, because along the whole approach, you will have invested a sound amount of time for finding a good value for each factor, but you will spend more time on the essential factors than on factors that play a very small role, which makes actually sense. So this is the general rule. In this strategy, you see that we speak about modeling, and it implicitly means, if not explicitly, that we have to deal with models, with mathematical models. And this is what I want to show now. In fact, if you take the example of the temperature, and we take a chemical process, maybe the temperature can vary in general, or in theory, from a very small value like 20 degrees to a very high value like 200 here. But of course, the impact of the temperature on the result would not be the same along this wide area. So since the purpose is to use a mathematical method that doesn't know chemistry, you have to be careful there. And instead of trying to do the experiments in this white rectangle, you have to choose at best small regions of interest, maybe having the temperature going from, I don't know, 40 degrees to 80 or 90 degrees here. And then you do in this small pink zone, you do your experiments. And once you have done that, what you may find is that the trend goes to an improvement in this direction. So as you then move to the next phase of the strategy, which would be, for instance, modeling, then you can change the zone and move it around. In this way, you will be able to combine the 3 phases of the strategy with a sound definition of small interest regions that enable the method to work properly, although it doesn't know anything about chemistry. So this is the background about the models. Let's make it more concrete by looking at the types of models that are used depending on the phase here. So I'd like to recall that we have these 3 phases, and depending on the phase, we'll use a different type of model. This is what we can actually see now. The several model categories, they are presented graphically here, and for screening we will use linear models. So you have to imagine that you have one parameter here, like the temperature, maybe another one here is the concentration, And vertically in the other direction, you will have the outcome of the experiment, which will be a yield like this, and it will typically be a plane. If I were drawing the same axis on the other ones, if you were doing modeling in a linear model with interactions, you could get typically a saddle like this. So, and then you see that at some point you get the best point in some corner, because if you walk on the surface somehow to increase the outcome, then maybe you start here and you can walk up like this and you'll end in a corner there. Finally, in optimization, we'll use quadratic models which can yield any kind of surface, as you see here. And in particular, we can get a hill or a valley like this, as we saw in the introductory example. So you can really find the best solution at the top of some hill-shaped surface. So just to show you that the idea is not to is to adapt the model complexity and the number of experiments depending on the phase of the strategy. Nevertheless, some of you may think, yes, but we vary all parameters at the same time, so how can we figure out what is the impact of each individual parameter on the result? And this is what I'd like to show now. How to estimate the factor effect. For this, I will suppose that we have two parameters called here A and B. You can think of concentration and temperature or anything of your context if you want. And we'll do 4 experiments here. This is Factorial Design, as it was mentioned in the invitation as well. But these are, of course, not the only designs that you can do with the software. Here we choose the 4 corners. So each factor can vary between its lower limit and its higher limit. And we put the experiments in the corners so that we can guess from these 4 experiments what happens in the whole square. And then you do the experiments and you get 4 results, of course. These are the green numbers. So now, if we want to find the impact of A, what happens if you increase A from low to high? And what you can see is actually for low A, the results are more or less about 30. And for high A, the results are more or less 70 or 80. So the impact of changing A from low to high is the difference from here left to right. And this, for the main effect, what we do is we take the average on the right, the average on the left, we make the difference, and we find that the effect of A is just 43. And then what we can do is to do the same for B. In order to make it visible for B, I'll change the color. And what I do now is I take B minimum here. So on average, the result is 50. And at the top, it's 50 something. And you compare again the difference between the bottom and low B and high B here. And if I do this, you get a difference of 8 only. So it shows you that the factor A, parameter A, let's say the concentration, is much more important than B. And this enables you to make a more precise fine-tuning of A than of B, because for B it may not be worth it. So this was about the factor effect. Let's look now at the interactions, since we mentioned at the beginning that interactions were important. Keeping in the same context or using the same yellow square, we can actually look at interactions. Interactions mean that the effect of one factor depends on the value of another. This we can see actually there. This is the same context, but I have just assumed that I get different results. And if I get different results, then I can see something different, of course. And now what you see is that if you increase A for low B, actually the value measured goes down from 80 to 34. But if you do the same change of A for high B, it goes up. And the fact that these two arrows go in opposite directions is a proof that there is an interaction between both. And there's some formula to calculate mathematically the value of the interaction, which is actually that one. Don't want to go here into detail, but it shows you that the strength of an interaction here can be as large as the strength of an individual factor itself. So having seen that, and this is probably the most complex mathematical slide of the whole presentation, I'd like to summarize how it works in general, because this was just explaining you the basic calculation to demystify the methodology. In general, if you have several factors, the software will calculate linear regressions to fit a model. So if we consider that we have only one factor and one outcome, so let's say this is the yield, and this would be, for instance, the temperature, and the yield increases the temperature, you have made some measurements here, and it will just fit a straight line like this to the point. And of course, if you use a quadratic model like we would use in optimization, actually you can fit a curve and it looks actually differently. The principle is the same. You fit a line like this. But of course, in order to fit a line, you need some points. And which points? How many points? That's the main question here, and that's the difficult point. So let's speak about the application of DoE now. So after these explanations, I hope that you took the information that it's not that complex, and provided that you apply it with a sound strategy. And then you want to use DoE with which design? Can you please mute the microphone? Which design use? So depending on the number of points, you will get a better or worse result. Of course, you want to calculate interactions if needed. You want to calculate the effects of all the factors. And of course, you want the calculation and the analysis to be reliable. And the main question, of course, for you is how many experiments, because this impacts on the work you have to do in the lab. So in order to do this, you have to choose the design carefully. So in order to achieve this, you can either go to take a statistics book and look at that in detail yourself, or the easiest way is to use a software solution. And this leads me to the next part of the talk, which is about the efficient implementation of design of experiments. So are there any questions about this? So if there's no question so far, what I would just a moment, please. I'll try to solve this background noise problem. Okay, should be fine by now. So let's go on with part 3 about how to apply DoE efficiently in practice. So in order to apply that efficiently, you as a scientist, shall you be a chemist, pharmacist, engineer, biotechnologist, and so on, according to us, you should concentrate on problem solving, because what you want to do is to solve your practical problem. You don't want to fight with complex mathematics or to have to understand all the statistical details possible. And especially what is important is that if you use a method like this, for which you may not understand all the statistical details or reasons for doing like this or like that, you should have a limited risk of using an inappropriate model. And this risk should be ensured, I would say, by the other side, which can be typically software or specialized consultants that can help you to avoid the traps by providing a guidance through the whole experimental process, providing a full analysis of the experimental results you will deliver, of course, and especially that provides you the analysis and interpretation of the results in form of reports that you can easily understand, especially with graphics as well. So, and also the software could tell you, as you go from one phase to the next one, which factors are important, which ones you should keep, and which ones you should eliminate. So if we put this in a graphical setting, actually the computer-aided work process would be the following. We have the user input first. This, you have to define your problem. Of course, no software and no consultant can invent the problem in your place. Then the software will support you for choosing an appropriate design based on the strategy, based on what you have input yourself. This design, you can use it, modify it if needed, and then go to the lab, let somebody make the experiments or make them yourself. And then after having input the results in the tool, you will get the statistical analysis and interpretation. Once you know maybe out of 15 factors which ones are relevant, the question will be whether to go on, and you will probably want to go on then into a next cycle. So you will go back to the top or to the left, I would say, and the user input, you don't have to do it again, because out of the list of 15 parameters, then the 7 important ones are now known also by the software, so you don't have to type them again. Everything would happen in the software in a same file, and then you start again with a smaller problem. What is written here as expert knowledge can be the knowledge of a human expert or as a consultant, or the knowledge that we have encapsulated into our DoE tool STAVEX to help users to solve the mathematical parts easily. So if you take now this procedure from left to right and you turn it by 90 degrees, so that the user input becomes at the top and the next cycle at the bottom, then you get exactly the user interface of our tool STAVEX. So it looks like this. You have 10 buttons from in the top-down approach. So you start at the beginning and you go down to work with it. The blue button is the one you have to use next, at least it's the recommendation of the software. And in fact, the principle is like this. You define the problem first by defining the quality attributes, the process parameters, material attributes, so-called response variables and factors in actually designer experiments. Then you get a good design. This is the purpose of these 2 buttons. You have to do your experiments, of course, and the analysis will help you to understand the result. And the next cycle, which you find here again, will help you to refine the analysis in another cycle. You see here at the top, it says here, so it's the beginning of a new project. It says here cycle 1 to 1, but you may have several cycles in the same study. So what does STAVEX for you in all these contexts? Actually, the advantages are the following. As you have seen with this top-down process, it will guide you through the whole experimental process from the problem definition to the choice of the design and to drawing the appropriate conclusions on the basis of the analysis. But of course, this guidance is also not stiff and not strict. If you have some practical difficulties, like an experiment didn't work, or you wanted to perform the experiment at a temperature of 40 degrees, but in fact it was 42, all this can be taken into account before you do the design or before you do the analysis, and so that you get correct interpretation at the end. The software will perform the calculations for you, the analysis, of course, and last but not least, it will write a report in HTML format in English with full sentences, so you can actually read the report and understand what goes on. For instance, which parameters are important, what's their impact, etc. And additionally, you get some graphics like this, contour plots or surface plots, and then you can actually look where the best point is on the basis of the color scale. Finally, the tool will help you to decide how to refine the results as you move from one phase to the next one. So to give you a flavor about the use of the software, let's speak about the users. The software is used now by more than 200 companies in the process industry, in pharma, chemistry, biotech in particular, to be more efficient in the lab and to find better results, of course, to save time, and also to fulfill the regulatory requirements. Examples of companies in life sciences that use this, you find large companies in the Basel region or in Switzerland, I would say, like CSL, Roche, Idorsia, or Novartis. And I've mentioned here, as these are just a few examples, I've mentioned here in red some Indian companies that use STAVEX as well. Like Arti or Robindo, Sanofi in India, and Zoetis close to Mumbai. Also, of course, the software is also used in fine chemicals. So there you find also large world companies like BASF or Givaudan in the flavor area, or Huntsman in the polymers. In India, Nosil is one customer there in Mumbai. And finally, in other areas, we have smaller lists of users in the food industry and also in automotive. And of course, engineering schools and universities use that both for teaching and also for research as well. So we've spoken before about the importance of this three-phase strategy, which I'd like to remind you here. So this was that here, adapting the complexity to the problem size. And then we have seen that there are several types of models as well. And we've seen that STAVEX goes over several cycles here. So I'd like to explain you the matching between these two concepts. And this is what comes next. This is the combination of the three-phase strategy with the STAVEX cycles. So screening the purpose is, of course, further to eliminate the irrelevant factors. It's recommended to use that by the software if you have a large problem. Large means more than 8 parameters. It then calculates a linear model and analyzes the importance of the factors with the so-called half-normal plot, if you have already heard this concept maybe. For modeling, it is rather recommended for problems of intermediate size with 4 to 8 parameters. You can calculate interactions, and then you perform classical regression with contour plots. Then you get these colorful graphics that are very helpful for the analysis. And finally, in optimization, it's the remaining zone, so less than 4 factors, quadratic model, and again, contour plots to find the top of the hill, as we have seen in the introduction. So this completes the part 3. Are there any questions? Yeah, there were a few questions, so I'd like to answer them both in writing, and so the data points are so the data points are chosen on the basis of the factor range definition. And you don't need to have initial data So what you may need to do, you don't need to do, but if you have done some preliminary experiments, you may want to integrate them into the design. But normally, you need, the design is only based on the definition of the factors and of their ranges, and not on previous experiments. So you could start with a new problem, and if you know more or less the ranges, if you have good inspiration for that, then you can do design directly. So far for the questions, are there any other questions? Then I see one. Okay, thanks. So let's go to the next part then, which is even more application oriented. It will be about case studies presenting. I'd like to present 2 maybe, and then I'll switch to the software demonstration to make it yeah, there's one more question, just a moment. So, if there are no more questions about this, let's speak about the applications. So first of all, I'd like to present a list of applications. Here we have done very many applications in the process industry. What we have here is actually process R&D, many applications there for synthesis optimization, biosynthesis optimization, should be for product quality, for process yield, typically for API process R&D scale up, also speeding up crystallization or filtration processes, optimization of grinding processes, all the typical processes of the chemical part or of the pharmaceutical industry, for instance, can be considered there. We have also much work on formulation development, like formulation of tablets, analyzing the dissolution profile of long-term release tablets, and also outside the pharmaceutical industry, formulation of cleaning products, formulation of PVC products, and what in the pharmaceutical industry I'd like to mention as well, formulation of injectables, so it must not be tablets, it worked also for liquids, with some users there as well. If we leave the R&D area, there are also applications closer to production, like for process validation, for instance. We have optimized the process for blister thermosealing for the pharmaceutical industry as well. And where one may not think of that as well is analytics for HPLC method optimization and robustness analysis for HPLC methods. One can use this as well. So besides the list of applications, I'd like to show you a few. Just answer one more question, then I'll go forward with the presentation. So after having said this list, let's go on with an example of synthesis process optimization here. And I'd like to present you this application, which was related to a quality drop in a running production process. So actually the process was already running, so it was a critical situation. The objectives were obviously to identify the root causes of the difficulties, to tune the process as well, and then there were, of course, some constraints. One wanted to solve the problem efficiently, so it should not take too much time because it was critical for the production. So there was a time limit of 4 weeks for the experimentation, and of course a financial limitation as well. So knowing this and knowing the cost on the time needed for each experiment, you can calculate easily how many experiments can be done, and this is done by the software as well. The most important part, of course, is not that, but it is what is to achieve. So since the quality was the major problem, actually it has been decided in this problem setting to consider a quality standard. So the ideal quality one should get, and to calculate as a response variable, so as a quality attribute, how far from this ideal solution or ideal quality one was, and to formulate this as a number, which is the deviation. So deviation of 0 means everything is fine, and a deviation of 5 is worse than a deviation of 2, of course, whatever the criterion shall be. And next to this, since it was synthesis, actually you have to do the yield, and the yield must be maximized because nobody wants to get good quality and a low yield. And of course, if the people were asked about the most important, since it was a quality problem, so the most important variable was the one related to the quality. So in this problem setting, I had a few questions just recently about how to consider which factors to define and so on. People did brainstorming about what could play a role. And I can assure you from experience that if something goes wrong, everybody has a good idea what should be changed. And this yielded a list of 11 factors that you can see here. So we have classical factors like the reaction time, the concentration, here, the temperature, maybe some waiting time, etc. So all these are numerical factors, as you can see here. And so you can take any value in these intervals. And besides this, we have discrete factors as well, like the shape of the steerer. You can have a propeller of an anchor here, or the ambient temperature, which can be cold or normal. And these could have, for the sole supplier, actually 3 providers to choose from, for instance. So it's a very complex list, I would say, of many factors. You could have even more if needed, but this gives you a feeling that it's possible to deal also with complex applications. And based on this, the strategy has been applied. So I'd like to recall what we said before. We said the strategy has 3 steps, and in STAVEX, it runs like this. It tells you if there are more than 4 factors here, then one should apply the screening, which is the initial phase of the strategy. So if we look at the application of this strategy for this example, we actually get that 11 factors. Screening is easy in terms of calculation, so the number of experiments is more or less the number of factors. So this is why if you have like 2 factors more, it's better to add them because it costs only 2 experiments rather than assuming that these factors don't play a role at all. Then we found in this application that only 6 factors were important. And with the 6 factors, we were able to reduce the complexity and thus to go to the next phase 2, which is modeling with 8 experiments here, a little more experiments depending on how detailed you want to analyze that in terms of interactions in particular. And in the modeling phase, one found that 2 factors were essential, these concentration and temperature. We've seen that actually in the initial slides. It's based on a similar example. And the optimization had 9 experiments. So you can see the difference depending on the phase. For screening, here we have 12 experiments for 11 factors. And here we have 2 factors and we have 9 experiments. So this is why you cannot do the fine-tuning at the optimization level with 11 factors, because you certainly don't want to do hundreds of experiments to analyze things that don't play really a role. But if we consider that overall, actually, what you see is that we had 11 factors here. What we have done are 12 experiments here, 8 there, so 20 in total, plus 9, 29 experiments to get a lot of knowledge about the important factors, about their impact, and to find a good solution leading to actually a quality deviation of 0.6%. So after all, you get a contour plot like that for these important factors. The blue points are the points where the experiments were made with the experimental result obtained. And the colors, they correspond to the quality deviation, and the best point is actually this one here at 0.59. What you can see in this plot as well are the interactions. If you increase the concentration here for high temperature, the colors don't change that much. Keep in mind the colors represent the value of interest, so the quality attribute. And actually, if you do the same concentration increase for low temperature, the colors change a lot. And this means that you have to pay attention to the concentration value, in particular where if the temperature is low. If the temperature is high, it's not that important. So I have another example here. It's using a lab reactor. As you know, the products from HEL are like Polyblock, for instance. So there are, you can optimize response variable, whatever it is, and vary factors. So if you have not that many factors, of course, applying the strategy, you don't need the screening. And you can apply the second phase immediately. So you can start with modeling as your first phase. Then you find important factors, and finally you get the optimization. Optimization with 3 factors takes, of course, more experiments than with 2. And finally, one gets the results here. I'd like to stop with this slide presentation for the moment, and what I'd like to switch to is actually to the software demonstration here. So what I'd like to show now here is actually the software. So this is what you have already seen in the slides. So this is the main window. And what I'd like to show you is how to proceed in practice. So the first buttons here, they are for defining the project name. They are not so important here, so I will skip them. So it's called new project by default, but you can change the name, put any remarks. It's just for documentation purposes. And it tells you in blue what to do next. Response variables must be entered. So I just click on the button response variables here, and I get a window in which I can enter, make it smaller, I can enter here response variables. So you can start typing in this empty table. So I'll put, for instance, the quantity of impurity 1. Then I can put the total impurities, and I can put the yield just to make a standard example. Then you can, of course, put the unit here as well. So all this is in percent. I will just put it here to make it properly. And the response type is quantitative. You may choose also qualitative if you are measuring something visually, like the color of some powder here. So usual case is quantitative, of course. And in this column, range and optimization, actually you have to choose between the optimization directions. And you have, these are buttons, and 3 are at your disposal. So if you click on actually max here, you can choose whether the impurity shall be maximized or minimized. Obviously, minimized is the solution. And then you can also choose here target value. You can say, okay, the target value is 0, and an upper limit for impurity 1, maybe 1, for instance, or 0.5, whatever you have in your specifications or in mind. And then click on this OK to be on to complete that. For the total impurities, I would say I put minimum again logically, and for the target value I'll put 0 again, and for the upper limit I would put maybe 2.5 here, and for the yield it must be maximized, which is the default value. I would say the target value is maybe well, let's put 90 something, not 100. Let's put maybe 98. So if you're happy with 98 already, and I would say the lower limit might be 85. Below 85, it's not acceptable, something like that. And with this, you have already defined the settings. And you can choose with this button which response variable is the most important. I would say it's impurity 1. It's the first one. It's the default, and this is why this button has become yellow. Then we click on OK. We assume that this is well enough defined. And we go to the next step. And the next step is defining the factors. Note that the button has become blue to highlight again what you have to do next. So this is this guidance I was speaking about before. So if I click here on the factors button, you have a similar table where you can list the factors, and you can list actually the regional values of the factors. So I will put classically the temperature. Then I put here the reaction time. So of course I use classical examples of synthesis, but you shouldn't get the feeling that it works only for that. So there are many applications in many areas. The factors use standard factors so that they are widely applicable and ensemble to everybody. So I would put here the stirrer speed, and I can put some concentration as well. And I could also put a stirrer type to stick to the example before. So I'll put the units here to make it sound. Steer speed, I'll put RPM, concentration in percent. The type has no unit, of course. And then for the factor type, you have here, all this is quantitative, of course. There are many types of factors, actually 4. Quantitative, qualitative, which we need now for a stereotype, mixtures for formulation problems, and we have also alternative factors that are used for solvent selection problems of particular kind. I put here qualitative, and I'd like to point out that in the software you always have some comments that help you to use it properly. It tells you the region is insufficiently specified, and for instance, please give a region for that, etc., etc. So now the only thing remaining is actually to look at this region of values here and to define values. So for the temperature, I can say the temperature goes from, I don't know, 30 to 50 degrees, and you can put an increment of 1 if you want to set only integer temperatures. For instance, tells you the values 30, 31, and so on are possible. Of course, it doesn't mean that they will all be tried. Then I have this. I have to do the same for the reaction time. I'll put 10 to 20 minutes. I can put it directly like this. The increment is optional. The stereo speed, I can put, I don't know, 100 to 200 maybe, with an increment of 10. Here, the concentration, let's put between 2 and 5%. And for the stereo speed type, since we have put here a qualitative, then I can click here and I can put 2 values, let's say A and B. For that, you can put up to 9 levels here. And then we have this, and all the messages have disappeared. You can click on OK, and the problem is defined. I may get a question a little later about this level change. You may wonder what that means. That indicates whether it's easy for you in the lab to change the value of a factor. Changing the temperature is usually easy, or changing the speed of the stirrer is usually easy. You turn a button and that's it. But changing the stirrer is much work, because you have to get one stirrer out of the machine and put another instead. So then you can put here hard, and then it will present you the experiments in a particular order, so that you don't have to change the stirrer that often. And then I click on OK, and this problem is actually defined. I will save the file here. And okay, so I have here this file which is saved, and now I come to the design plane. So now we have defined the problem. We have actually to choose a design. So an important point is that the software tells you, according to the strategy, which phase, whether screening, modeling, and optimization, is the best one for your problem. This is the STAVEX option here, STAVEX modeling. This changes dynamically depending on the problem you enter, from STAVEX screening to STAVEX modeling or STAVEX optimization. Of course, as a user, you can choose that yourself by using this button. So if I am in modeling, it tells you to do this design or this design or this design. These are the best ones. So the top one is always the best one. So if I click on it, it gets calculated. It tells you it's a full factorial design here, and it's the best design, and some comment about the calculation of interactions, which is possible, which is actually good. The second one, you may want to calculate it as well. It tells it's second best. It's particularly interesting because it has only 16 experiments instead of 32, and so on. If you go to user optimization, you'll get a bad surprise, namely that, of course, you want to look at this rather large problem in more detail. So if you want to go this way, you will have to do more experiments, as you see here, of course. So we don't want to do that. So and if you go for screening, it's more quick and dirty for this problem, then the number of experiments is much smaller. So having done this, I can click on OK. And for the choosing one of these green designs, I will, for instance, choose the second one, this one. Here, what you notice is that the stereotype here is on hard, as we said, and you get all the experiments with A at the beginning and all the experiments with B at the end, and it tells you have to change the stereo only once. So this is convenient for the lab. Each row here tells you actually for each experiment, experiment 1, experiment 2, experiment 3, which setting of the factors you should use. So experiment 1, a temperature of 30, reaction time 10 minutes, stereo speed 100, concentration of 2 with stereo A, and so on and so on. What you can do easily is to actually here export this into you can export this into Excel. Let me add here, take a white zone. And if I paste this, actually you have already here the design that appears. If I take this here and I make it bold, then you get the design. So you can give that to somebody in the lab who has not the software or who wants to have it in Excel, and then you have actually the design here, 16 experiments to be done, and you can collect the results here from the, as you make the experiments in this zone, and as you input something there, you can copy this green zone into the software itself, not anymore there because this is the design, but in the next step, and the next step would be actually logically the results that are marked blue again. So normally I would go there, you have the design at the top, and you can see here, actually if I want this experiment, it's the fourth row, and you can enter there directly your experimental results, of course, but you can also enter that, input that from Excel. So there was a question about multi-level factorial designs. I'd like to, I cancel that and I can show you that here. So if you go to user optimizations, you have these designs here, 3-level full factorial designs, for instance, that are used. You have many types of designs. It just depends on the phase you are using. So for those of you who know the design types, the modeling is more or less factorial designs. The optimization are response surface designs like Döhler, central composite, Box-Benken, multi-level full factorial. And then in screening, typical design would be a Plackett-Berman design or super saturated design. I don't want to bother you with too much design theory here. And I'll go back to the demonstration. I hope I answered the question. So there was again a question about the agitator type. It's what we've seen before. So it's here the stereotype. So if you have two stereotypes here or agitator type, you can put 2 or 3 as you want, and you can consider that the impact of the selection of the agitator as well. So now I go back to the results. As you can imagine, for this invented problem, I don't have any results, but there is a magic function for demonstration purposes that enables one to generate results. So the experiments have been magically done here. And what I do here is actually to complete this, and I'll try now to perform the analysis. Of course, the results are invented, so the results might not be good, and if they are not good, I'll grab to a standard example. Yeah, this might not be too bad. Yeah. So let me check about the models here. This is bad, and for the yields, yeah, it's bad as well. So let's take maybe this one. So we had actually 5 factors. It automatically tells you which are the important factors because we are in modeling. So it finds out of 5 factors, 3 are important, distinguishing between those that are really important, which are called probably important here, because in statistics you're never sure of anything, and the possibly important are the half important factors, I would say. It tells you the model equation saying that if you know the reaction time, the zero speed, the concentration, then you can calculate the value of impurity 1 for any value of these 3 factors. Then it makes automatically below that model diagnostics. Of course, this model diagnostics tells you that the fit is poor here because I invented the results, and with invented results, of course, you can get whichever result quality you want, but at least it checks for you. It shows that it checks for you all kinds of statistical constraints and tells you whether it's worth or whether it's fulfilled or not, saying no, yes, or questionable. And if it finds a better solution by making a transformation, it suggests you to do it with a log transformation here. So if I want to go to the details, yeah, sorry, that was not what I wanted. If I want to go to the yeah, that was it. I just want to show, so this works much better. So what I'll do is this is an opportunity to show you how it works with self-optimization of the models by the software. So I can go here to the next cycle and perform these transformations suggested. And then you come in a new cycle. You don't have to do any new experiments there. And then you get actually a better fitting model. So with other important factors here, and after all, you get something which is very good. So it tells you as well that the best way to display the function is the contour plot. So what I can do is to make a contour plot for the impact on these impurities. I'll take a few factors, 2 factors to start with. I can represent the impact of these 2 factors on the quantity of impurity 1. And I get a plot like this, which shows that actually you can also read the values directly there with the mouse. If you go with the mouse, you see above the color scale on the right, there are a few numbers for the temperature. And if I move the mouse, the temperature and reaction time values change, and the impurity 1 changes as well. The design space can be obtained at least for this response variable, because you see that in the bottom right here, it's all dark red, and dark red means it's out of specification. And if it's out of specification, actually, yeah, it doesn't fulfill your objectives, of course, for that. This is just an example of plot you can make. If we want the same plot for the two agitator types, we can get that as well. And you see that the colors of the rainbow zone changes and that there is more out of spec with stirrer A than with stirrer B. So this enables a three-dimensional analysis of the situation. And we can go on like this also in 3 dimensions. So if I put now stirrer B only because it was better, and I take the stirrer speed on top of that, I can represent the impact of the 3 factors actually on the impurity 1. And there is a tool here that enables you to increase the stirrer speed and to see what happens. And you see for high stirrer speed, you get more out of spec zone. So that's why it's profitable to have a low stirrer speed here. So far, for giving you a feeling about the software functionality and what you get as well, of course, and this is important, is the best level combination here in this report. It tells you which factor settings are the best ones, which amount of impurity 1 you can get then, and it's occurs in modeling and optimization for that. So what I'd like to do now is to ask you whether you have any questions about demonstration that have not been asked yet per So I will answer. Sorry, are there any questions either orally or per chat? So the question I get here is about the joint impact of 2 continuous and agitator type on the results. So what we want to do is the impact of temperature and time. And you cannot put the stereotype of on an axis because the agitator type is not a continuous factor, but you can make the same representation of the impact of temperature and time on the impurity for different stereotypes. And if you do this, then you get 2 plots that share a common color scale. So the color scale represents the impurity 1. And here you have stirrer A at the top, and here you have stirrer B. So since the color scale is common, you see that the best values, the red zone here, the low impurities, are achieved with the red and the yellow. You find them only for stereo B, but not for stereo A. And you find then the impact of temperature, reaction time, and the agitator selection on the impurities in a single combined plot. And moreover, the out-of-spec zone is in dark red in both. I think this was the question asked before. Somebody wants to show to see the slides design choice for screening, modeling, and optimization. I guess it's not the slides, but the software. I guess this one. Okay, so there was a discussion, there's a question about so I'll close the plots for the moment. Yeah, I can leave the plots here. If you go to, actually, I'll go to cycle 1 maybe here, get back here. And so the question is about this here, modeling, screening, etc. So actually, the screening designs, they correspond to the screening phase, as I said. So they are used, these are easy designs, so easy to realize, small number of experiments, impossible to analyze interactions because they are too small for that. The main advantage is that they have little experiments. And so they are of major interest if there are many factors and you don't know which ones are the relevant ones. Because then you can quickly sort out which ones are relevant and which ones were just good ideas of your colleagues, but in fact are not so good ideas. If you go to modeling, then that's the intermediate zone. The top design is always, the best design is the first one, and so on. I'd like to mention as well, you have also a so-called external design, which is an empty table that enables you to import any data you want and to use the tool only for analysis purposes. And or also to add some preliminary experiments. If I click here, you get an empty table. You can enter whatever you want, maybe 2 experiments, and let the software calculate the remaining one. You have in this modeling phase, which is the intermediate one, you can influence which interactions you want to consider. So interactions between the parameters, you have the 5 factors here, the 5 factors there, and it tells you fewer interactions is fewer experiments. So if I suppress these interactions, or if I put only 1 here or these 2, and I click OK, it will recalculate the designs. And if I click on accept here, I get a new list of design, and like a miracle, you see that the number of experiments has been divided by at least 1.5, because each interaction is like an additional factor, and then you need experiments to be able to calculate them. So this is what you can do at this level, and for optimization, you have more details, so you have to do more experiments, and this is why one does that only if the number of factors is already limited, thanks to the preliminary phases or thanks to your initial knowledge of the application. Yeah, hello. I just stopped the audio because I wasn't speaking because I was answering the messages, and I suppressed the audio so that you don't use the typing. I suggest I continue with the presentation, and I will answer the individual questions after that. So actually, the presentations, the presentation of the slides is well advanced. What we have, let me start it here again. So we've seen that already, and we had the demonstration as well. So what I'd like to show here further to this is how this can be used for quality by design and the design space. What you've seen in the software demo is actually that you can mark in dark red the out-of-spec zone in the contour plots. And this is typically the kind of example you get. So the dark red is out of spec for this quality deviation example we had in the slides. And so the rainbow zone is actually the design space according to the QBD guideline. Of course, there are some people who still speak about fixing limits for the factors, which I call the traditional way. But if you do this, you notice that you have to set the concentration from 9 to 12 and the temperature from 40 to 45 point something. This is this red zone, this rectangle there, and then you lose something because since the design space is round, if you try to put a rectangle in it, you cannot take everything. So this was for the quality deviation, so for one quality attribute. Of course, you can do this for several quality attributes at the same time as well. And so this is the same picture we had just before. But if you do that for the yield as well, you have another picture, another dark red zone, which is the out of specification zone for the yield. And then what you notice is that the green zone, which is the same on the left, or the green limit, which is the same on the left on the right, has to be pushed inside due to the yield. And then the design space for both together actually becomes the orange border. So this zone here, all this zone here within the orange border is the multidimensional design space with regards to the two factors and the two quality attributes here. So this actually shows that the design space can be calculated actually using DoE advantageously. You get actually more process knowledge here. You get more flexibility in production because moving in a design space doesn't require, as you know, revalidation. And now many companies, they register design spaces as well. And so you know more or less which questions will come on that. So this can be applied in general, independently of the application, and even with complex problems, it doesn't change the strategy. It changes, of course, the problem definition, but not the way you work with it. So there are many further practical aspects. In view of the time, I would like to skip this. You will receive the presentation. So mainly we've seen the difficulty of changing the stereotype. For instance, you can say an experiment is not feasible. So I can quickly show you a few of those. For instance, you could say, without going into the detail, if you know that some of the experiments are dangerous and because it will explode and you refuse in advance some experiments, of course you can do that. And the software will adapt design for that. Then there's also the possibility to modify a design here if you have done some preliminary experiments or experiments done in the past already before you got the idea of using DoE. If you cannot stick in the lab to the values of the factors you should have stick to, you can do this. Of course, if experiments don't work for some reason, or if you want to do some adaptations to the design as you want, you are flexible to do so. There's a feature called in the software design customizer, and there's another one which I've shown you is this external design, which was the empty table for the design. Also, what is possible is to consider restrictions. So if you say something like this, problematic combinations, high pressure, high temperature, long reaction time, this combination is impossible. So you can define yourself some restrictions of several kinds and say so that the dangerous combinations are excluded from the design by the software itself. And finally, I'd like to mention as well that there's a possibility to take account not only graphically, as we've seen, of several quality attributes or response variables at the same time by using a so-called desirability function. This is an additional calculation that can be made by the software optionally, and once you have said what are the specification limits for the quality attributes, what is the relative importance of each quality attribute, then it calculates for you the best compromise setting so that you say, I have like 5 impurities, and what is the best parameter setting overall that decreases more or less all impurities at the same time. Then you get an overall conclusion about that, and not you don't get high quality but bad yield or reversely. So based on this, I'll come to the conclusions of the talk, as the time is almost over. So if you have thought at the beginning that design of experiments may be a burden or not helpful, we've hopefully seen in this presentation that it doesn't apply, but rather the contrary is true. There are many prejudices around about design of experiments, like too many experiments are required. The truth is actually that on average it requires less experiments and trial and error. Of course, nobody can exclude that you have a great idea and you do one experiment and this is the best solution. A second prejudice is that the process variation is too high to use statistics. Actually, statistics is the science of analyzing variation. So this doesn't really apply. An idea is that statistics is for data analysis, and actually statistics is more than that. So if you do the wrong experiments, actually imagine you don't change the temperature, you can do as many experiments you want, you will never find the impact of the temperature. This sounds obvious. This also works for interactions in a multidimensional setting. So data analysis cannot fix the experimental errors you do by choosing the wrong experiment. One factor at a time to avoid getting confused. I have shown you, I hope, in a second part that this doesn't work actually, and it's possible to find the impact of each factor even if you vary everything at the same time. And if you tend to consider that DoE means cumbersome calculation and results that are not understandable. I hope that the software demonstration has convinced you of the contrary. As a summary, I would say DoE enables you to become more efficient for process and product optimization. It is an essential component of QBD to provide the design space. And with our tool STAVEX, conceived as an expert system, and this guidance can be used very easily. This tool STAVEX, it has been designed and developed at the beginning in the pharmaceutical industry, so specifically for the needs of scientists like you. The guidance makes it user-friendly, and it is used by many that are not statistical experts and that take profit of the flexibility and the user-friendliness of the tool to go forward on that. This will be my conclusion. I'd like to thank you very much for your attention. If you want further information and get a trial version for download, you can test the full version for free from our webpage. And if you want information about the software locally, please keep in touch with the high-tech systems, which I'd like to thank as well for the organization of this webinar. And with this, you can get the information required. I just like to show you to finish our web page here. So if you go to download and here software downloads, then you get the software package to download, and you will get a license for 1 month with which you can really test on a practical application of yours. And if in this period you have questions, feel free to send us an email as well. So this will be the end of this webinar. And I will take some time further to answer the questions. So if you have any oral questions, you can actually switch on you can switch on your microphone as well. And otherwise, I thank everybody. And I will take some time to send you the answers as well.
So please do send some email, whatever the queries you may have, and we will get back to you with the answers. Maybe you can download the trial version, and we help you there. It is a simple software, and for the QBD, it's very important that you take the help of the software to get the design space, the real design space for the various processes. Thanks all for your time. I look forward to your email for supporting the project. Thank you.